Good day everyone, I'm Ray Alcoran Tabas and I will discuss about the cell reproduction including cell cycle, MPhase, mitosis and cytokinesis, and the last is meiosis. Let's first discuss about the cell cycle. The cell cycle is a process in which a cell grows and divides to create a copy of itself. Some organisms reproduce through the cell cycle and in complex multicellular organisms. The cell cycle is used to allow the organism to grow and to replace cells as they grow worn out. In animals, the whole cell cycle takes around 24 hours from the start to finish. Some cells, such as skin cells, are constantly going through the cell cycle, while other cells may divide rarely. If at all, neurons, for example, don't grow and divide once they are mature. As you can see, this is the illustration of the phases of cell cycle. There are the mitotic phase, the first growth phase, the synthesis phase, and the last is the second growth phase. The cell cycle consists three phases: the gap phase 1, growth and synthesis, the S phase is the DNA synthesis phase, the gap phase 2 is the preparation for division, and the last is the M phase, which includes the overlapping process of mitosis and the cytokinesis. Next, we discuss about the different phases of the cell cycle. The first phase of the cell cycle is the gap phase 1, which begins at the completion of the mitosis and the cytokinesis and lasts until the beginning of S phase. This phase is generally the longest of the first cell cycle, and it's quite variable in length. During this phase, the cell chooses either to replicate its DNA or to exit the cell cycle and enter a quiescent state. Next is the S phase. Replication of the chromosomes is restricted to one specific portion of interphase called the S phase, which is typically lasts about 6 hours. In mammalian cells, the start of S phase is the actual annotation of the DNA synthesis is take place several hours after the cell has committed to carrying out the DNA synthesis. So during S phase, each chromosome replicates exactly one to form a pair of physically linked sister chromatids. In animal cells, a pair of centrioles is also duplicated during the S phase. Next is the gap phase 2, which is the portion of interphase that follow S phase. Some cells can exit the cell cycle from gap phase 2 just as they can from gap phase 1. So the combination of these three phases is known as the interphase. Last phase of the cell cycle is the M phase, which includes the overlapping processes of mitosis and the cytokinesis. The mitosis divided into 5 stages: the prophase, prometaphase, metaphase, anaphase, and the last is the telophase. So the cell cycle consists of two main periods, the interphase and the mitotic phase. Next is the cell cycle interphase. Interphase is the period of growth and DNA replication between cell division. The three phases of interphase is the gap phase 1, which is cell increases in size, the S phase or the replication of DNA. Two sister strands of DNA called chromatids are produced. And the last is the gap phase 2, which is organelles double, new cytoplasm forms, all other structures needed for mitosis form. The second period of the cell cycle is the mitotic phase. Mitotic phase is the stage when a cell divides. Mitosis is the division of the single nucleus into two genetically identical daughter nuclei. These divisions involves two processes: division of the nucleus, separation of the cytoplasm, and the new nuclei into daughter cells. Mitotic phase is divided into two mitotic phases. The first mitotic phases contain 4 stages: the prophase, metaphase, anaphase, and telophase, and the second mitotic phase is the cytokinesis. So, let's first discuss about the prophase. So in the early prophase, the chromatids condense becoming the chromosomes. Nucleolus disappears. In these spaces, centrioles separate and start moving to opposite ends of the cell, and spindle begins to form. Next is the late prophase. The nuclear membrane fragments and the microtubules invade the nuclear area. The centrioles have moved to the opposite poles, and the next is the spindle is completely formed. So that is what happened in the late prophase. Next phase is the metaphase. In metaphase, the chromosomes are aligned at the metaphase plate. The centrioles move at polar ends and projects spindle fibers to connect each chromosomes. So the third mitotic phase is anaphase, which in anaphase, the paired chromosomes or the sister chromatids separate. Separated chromatids move to opposite pole. Partial division of cytoplasm begins. The last mitotic phase is the telophase. In telophase, the chromosomes are at the poles. Chromosomes uncoiled and turn chromatin. Nuclear envelopes reforms. And the last is the spindle fiber disappear. In the second mitotic phase, which is the cytokinesis, which occurs at the end of mitosis, in animal cells, a cleavage furrow separates the daughter cells, while in the plant cell, a cell plate separates the daughter cells, and the daughter cells are genetically identical. As you can see at the left side of the slide, cells return to interphase. The control of the cell cycle, the regulatory proteins called cyclins control the cell cycle at checkpoints. The gap1 checkpoint decide whether or not cell will divide. The S checkpoint determines if DNA has been properly replicated, and the last is the mitotic spindle checkpoint, ensures chromosomes are aligned at mitotic plate. And that's all. Thank you.